let's get going with the heart. So, when we look at the heart, guys, gals, many people since antiquity used to think that the heart was the center of reason. The brain was just this miscellaneous organ, almost vestigial organ to ancient anatomists and physicians. They didn't really know what the brain did, but they knew that the heart was the center of intelligence. They knew that the heart was the center of thought and emotion. It was an established scientific fact. I find it very interesting that established scientific consensus is oftentimes turned upside down. Just recently, the age of the universe was lowered by 2 billion years, just last week, according to a new calculation of the Hubble constant. So I just want to throw that out there. Just because something is established doesn't actually mean that it's set in stone. So as of right now, the heart is not the seat of emotion. It's the thing that pumps blood for us. And that's how we're going to talk today in our 2019 A&P class. So when we think of the heart, it moves blood from point A to point B, and it's a double pump. There's two primary circuits for the heart, the pulmonary circuit and systemic circuit. It refers to lungs. So when we think pulmonary, I want you to think lungs. We're going to talk about the pulmonary system or the respiratory system later on this semester. We also have the systemic circuit. The systemic circuit encapsulates lungs. Ms. Londrich and I were, over the summer, we were just chit-chatting while we were doing lab prep stuff for you. And one of the questions I remember asking Ms. Londrich was, what about blood supply to the heart? Because the heart has tissue. The heart needs to be supplied with blood. Is the heart part of the pulmonary circuit, the systemic circuit, or is there a third cardiac circuit? So I did some digging, and since the major blood supply to the heart comes off the aorta, the heart itself is part of the systemic circuit. So that's just some interesting trivia for you. I like and I dislike this figure. I like it because it's kind of pretty looking. It has great colors. It's from your textbook. So those are all good things about the figure I include in this PowerPoint slide. But there's something horrendously wrong with this figure that perpetuates a common student misconception. And it's the idea that one lung receives deoxygenated blood, and the other lung will expel the oxygen. If you look at the figure, I'll highlight what I'm talking about. If we really wanted to make this figure correct, we would have deoxygenated blood going to both lungs, and then oxygenated blood leaving both lungs and heading back to the heart. In other words, we would make it look more like this figure that I pulled off the internet for you that shows deoxygenated blood entering each lung separately, and then while that blood is in the lungs, it picks up oxygen and then goes back to the heart. So this figure is technically more accurate, but it's certainly not as pretty as the one on the other slide. When we look at our heart, we have two sides to make up the double pump, the left side and the right side. When we look at our left side, it is going to receive fully oxygenated blood from the lungs. That fully oxygenated blood from the lungs is going to enter the left atrium. From the left atrium, it goes to the left ventricle. From the left ventricle, it goes to the aorta and the rest of the body with the systemic circuit. When we look at the right side of the heart, it receives deoxygenated blood from the rest of the body, from the systemic circuit, and that deoxygenated blood enters into the right atrium. From the right atrium, it goes to the right ventricle. And from the right ventricle, that deoxygenated blood is sent to the lungs, where it picks up oxygen and drops off carbon dioxide. And then from the lungs goes back to the heart. So we have a double pump to the heart. For those of you who do any kind of comparative anatomy or zoology classes with lots of dissections, some hearts are two-lobed hearts. Occasionally we could have a three-chambered heart, a two-chambered heart. Human hearts or mammalian hearts are a little bit more complicated. We have four-chambered hearts for the vast majority of mammals. Now when we look at the heart, there's some nuts and bolts stuff we need to talk about. Where's the heart located? Last semester we talked about body cavities. We can have the thoracic cavity. Within the thoracic cavity, we have the pleural cavity and the mediastinum cavities or mediastinum cavities. Our heart is technically, if we want to be really pedantic, it's in the mediastinum cavity. Media for middle, stinum for the sternum. It's in the middle, directly beneath the sternum. It's a common misconception among the general public that the heart is off to the left side and not in the center of your chest. The heart is centered within the mediastinum cavity, but it extends to the left. So as we look at our heart, it's right in the middle of our thoracic cavity with an extension to the left side of the thoracic cavity. And we'll talk more about this with the respiratory unit. That extension to the left side means that there's going to be some differences between the right lungs and the left lungs, just to accommodate that extension. Now, the apex of the heart, this is going to be a lab objective. The apex is the pointy end of the heart. And in human beings, the apex of the heart is going to be directed inferior. It's going to point down to the ground when we're standing in anatomical position. The base of the heart is the broad, wide end of the heart. And it's kind of hard to see. It's not very clearly defined like it's on the lungs or some other structures and organs in the body. So I'm going to draw a picture of the apex of the heart. The apex is right here. It's that pointy end of the heart. And then the base of the heart is right here. If we drew an imaginary line that chopped off all of those major blood vessels, that would be the apex, the base of the heart would be that straight line on the superior or top margin of the heart. A good rule of thumb for the size of your heart is to make a fist. And if you make a fist, that's about the size of your heart. Bigger people have bigger hands and make bigger fists, and we'll have bigger hearts. So it's just, just a general guideline, but to put some numbers to it, our heart weighs a little bit less than a pound, or about 10 ounces in a typical average adult. Now, wrapped around the heart, we have this double membrane. It's one of the serous membranes we covered last semester, the pericardium. And like all good serous membranes, it's a double membrane. We have two layers. We have the visceral layer directly against the organ, and we have the parietal layer against the cavity wall. So we have the parietal pericardium, that's the outer layer, and then we have the visceral pericardium, that inner layer. The visceral pericardium is the smooth layer directly on the surface of the heart. It is the exact same thing as the epicardium. So depending on our frame of reference, if we're talking about layers of the heart, it's the epicardium. If we're talking about the serous membrane, we call it the visceral pericardium. But in reality, it's the exact same tissue in the exact same spot, just renamed depending on our frame of reference. Now, in lab this week, we have a couple pork hearts or pig hearts for your learning pleasure, and I really hope you take the time to go up to the front of the room and grab the parietal pericardium with your fingers, and I just want you to tug on it just to see how tough it is. It's kind of cool to see how unbelievably tough this fibrous tissue is that encapsulates our heart. And when we look at this pericardium, it does a couple things for our heart. It helps to lubricate the heart for us. As our heart is beating in our mediastinum cavity, there's lots of movement to that heart. And as that heart is moving around, we need to lubricate it. And the lubricant is going to be in between the two layers of the pericardium. It also helps to control the size of the heart. It gives the heart a little bit of room to expand, but it also limits how much the heart can expand. Every once in a while, we find that there's going to be an inflammation of the pericardium. This is known as pericarditis, and as you can imagine, it's just very painful. Now, as we look at, we're changing perspective now. So on this slide, we're talking about the serous membrane, the pericardium. On the next slide, heart wall, we're going to talk about layers of the heart. So when we look at layers of the heart, instead of using the root word pericardium, we're going to use the root word just cardium. We have the epicardium on the surface of the heart. We have the endocardium on the inner lining of the heart, lining those chambers and cavities on the inside of the heart. And as we look at the endocardium in lab this week, I want you to go up to the front of the room, and I want you to feel how unbelievably smooth the endocardium is. This morning, Ms. Lundrich and I were slicing up some hearts for you, and you can go up and feel how smooth they are. As we look at the endocardium, it's worth emphasizing that this endocardium of the heart is a continuous lining with the inside of our blood vessels. All the blood vessels of our body are lined with a very smooth, very slippery surface. And this is to minimize clotting. And then in between the epicardium and the myocardium, we have the myocardium. You may recall myo is one of the root words that means muscle. We had myo, mys, and sarco. So our myocardium is the muscular layer of the heart. Our myocardium is going to be made of cardiomyocytes, or heart muscle cells, if we were to look at the root words of a cardiomyocyte. And as we look at the fibrous orientation of the muscle tissue within the myocardium, we find that there's a twisting appearance to these fibers. If you were to take the heart and slice the apex of the heart off, and then look at the apex of the heart just with your naked eye, you can see what's known as the cardiac vortex. And no, the cardiac vortex is not going to be on your exam, but it's just kind of cool. You can see how the fibers are spiraling towards a central point as they move towards the apex of the heart. And this cardiac vortex and this spiral arrangement of the cardiomyocytes makes it so that when the heart contracts, it's like wringing out a rag. For any of you who've ever washed a kitchen sink with a wet dish towel. And this wringing out of the rag helps to squeeze as much blood as possible out of the chambers of the heart during contraction. We also are going to have a fibrous skeleton of the heart. This fibrous skeleton is going to have a lot of collagenous fibers and is also going to have a lot of elastic fibers. So I'm circling some of our fibrous skeleton of the heart right here. This fibrous skeleton is going to do two key things. First, it provides a framework for the cardiomyocytes, the heart muscle cells. There's been some very I personally think exciting research that has taken a pig heart, and if you take a pig heart and dissolve all the cardiomyocytes off, you can have the fibrous skeleton left over. And then you can take a human an embryonic stem cell, put it in the fibrous skeleton of the pork heart, and then grow a human heart that's custom tailored to the genetics of the recipient. Now, of course, this is at the lab stage, the research stage. This has not been transplanted into a human being yet. As of 2014, custom tailored kidneys have been grown that were 20% efficient at filtering blood. So, knock on wood, within our lifetimes, organ transplant shortages will be a thing of the past, optimistically within 15 to 20 years. Now, as we look at the electrical insulation of the heart, I need to really emphasize this aspect of the fibrous skeleton. We don't want the entire heart to contract at the same time. There needs to be some coordinated conduction of the action potential throughout the cardiomyocytes. And this fibrous skeleton helps to insulate one chamber of the heart from another chamber of the heart so that all of the chambers don't contract at the same time. So in addition to giving structural support, it also gives electrical guidance as the action potentials are moving through the heart. As we look at the heart, it's important to know the chambers. I'll say this again, I've said it already, but the human heart has 4 chambers, 2 on top, 2 on bottom. The superior chambers, the one on top, are called atria. The inferior chambers, the one on bottom, are called ventricles. And we also are going to have left and right. Me personally, as a student and still as an instructor, I struggle with left and right all the time. It's a pretty common thing to struggle with. So we need to remember we have a patient-centered point of view. So if I'm looking at the heart and it's on my right side, that means that it's on the patient's left side when you're looking at it. But one of the common issues with hearts, particularly mid-sagittally sliced hearts or transversely sliced hearts, is it's hard to know, well, am I looking at an anterior view? Am I looking at a posterior view? So I'm going to give you a big hint right now. Look at the thickness of the myocardium. In lab, this is how you can differentiate the left and right sides of the heart. So when I look at the myocardium, The myocardium of the left ventricle, I'm going to put left V for left ventricle. The myocardium of our left ventricle is very thick. The myocardium of the right ventricle, we'll put an RV there for right ventricle, is super thin. So, if we just look at the thicknesses of the myocardium, as you can imagine, the thicker myocardium has more heart muscle cells in it. It can contract with more force. The left ventricle pushes blood to the systemic circuit, the one that has more tubes. The left ventricle needs to squeeze harder because the blood is traveling farther when it leaves the left ventricle. The right ventricle, on the other hand, sends blood to the lungs. And because the lungs are smaller than the rest of your body, the right ventricle doesn't need to work as hard and consequently won't have as many cardiomyocytes compared to the left ventricle. This is a classic example of structure determining function. The left ventricle is thicker, therefore it can function to move the blood farther compared to the right ventricle. Now, if we look at the superior margin of the heart, it is going to have the atria, the left and right atria. And these are going to receive blood as it comes back to the heart. The right atria will receive deoxygenated blood from the superior and inferior vena cavas. And then the left atria is going to receive blood from the left and right pulmonary veins. And as these atria are receiving blood, there's this little overflow chamber. Every once in a while, there's going to be a slight abnormality within the cardiac rhythm, and we have a compensation factor built in as we're moving the blood through the heart. These compensation factors are known as the auricles. And if we need to, we can use the auricles of the heart to enlarge the size of our atrias. In one of those situations, we're having a slight alteration to the cardiac rhythm and a slight temporary irregularity to the flow rate of blood. This figure does not do a good job of showing the auricles. Let's see if I can back up a couple slides here. Yeah, that's a terrible picture of the auricles as well. You'll see it in lab. Well, actually, we can see the auricles right here. If we look on the anterior side of the heart, we have the auricle right here for the right atrium. I would always tell my students that auricles have a scalloped appearance to them. The auricle for the left ventricle is primarily going to wrap around to the posterior side of the heart. The right auricle is colored blue because it receives deoxygenated blood. The left auricle is colored red because it receives oxygenated blood. I made sure to ask all of my students this question. It was one of your PhysioX review questions last week. What color is oxygenated blood versus deoxygenated blood? Oxygenated blood is bright red. Deoxygenated blood is dark red or maroon red. Deoxygenated blood is not blue. And that's a really common misconception among undergraduate students. The data shows about 20 or 30% of undergrads think your blood turns blue when it loses oxygen. So I want to make sure to be crystal clear. You do not have blue blood. Now, in between our atria and ventricles, we are going to have a slight indentation known as the atrioventricular sulcus. This AV sulcus is a indentation. Think back to the nervous system. We had sulci and gyri on the brain. A sulci on the brain was a shallow indentation. On the heart, we also have sulci. The atrioventricular sulci is a small indentation in between the atria and the ventricles on the surface of the heart. We also have the interventricular sulcus. The interventricular sulcus is in between, inter for in between. What is this shallow indentation in between? It's in between the left and right ventricles. As we look at our interventricular sulcus, on the anterior side, it extends down right here, and I really wish I could make this menu go away. I don't know how to do that. Yep, it's not going away. Sorry about that. The posterior interventricular sulcus is on the posterior side of the heart and extends downward in between the left and right ventricles. And as we look at these sulci, there's three primary things the sulci on the surface of the heart contain. They're going to contain adipose tissue. They're also going to contain arteries and veins. Arteries, veins, and fat, or adipose tissue, are going to be in the sulci. Now, if we were to take a transverse slice of the heart and to look at the inside of the heart, we have interatrial septum and interventricular septums. A septum is a dividing layer of tissue. So when we look at an interatrial septum, it is a wall of tissue, septum referring to a wall of tissue, that is in between the atria. Interatrial septum, meaning between atria, wall of tissue. On our heart, the interatrial septum on this diagram is right here, kind of that green triangle I just highlighted. We're also going to have an interventricular septum, a wall of tissue in between the ventricles. The interventricular septum in this diagram is being highlighted or circled in green right now. We're also going to have some little ridges on the inside of the chambers of the heart. These little ridges of muscle on the inside of the heart are referred to as pectinate muscles, and we have many pectinate muscles on the inside of our heart. As we look at the inside of the heart, I can see that there's a ridge here, there's a ridge right here, there's another ridge. There's many, many ridges on the inside of the chamber of the heart. Each individual ridge is referred to as a pectinate muscle. We're also going to have trabecular carnae. These trabecular carnae are the ridges that are present in the ventricles, and these trabecular carnae are going to lead up to some small, stringy-like pieces of connective tissue. For those of you that are a little bit more old school, the trabecular carnae used to be referred to as cardiac papillae or papillary muscles. That's how I was taught it back in the day. Your textbook uses the term trabecular carnae. But if I slip up and start saying papillary muscles, that's because that's how I had to learn it. Now, as we look at our valves in the heart, we have 4 chambers of the heart. We also have 4 valves in the heart. Our valves in our heart function to keep blood flowing in one direction. And this is very important, because if our blood starts flowing in 2 directions, then we can't efficiently oxygenate or deoxygenate our blood. We can't use our blood to reliably deliver nutrients and remove waste products to the tissues of our body. If we have a malfunction of one of the valves of the heart, the blood will start to flow backwards and all kinds of bad things will happen. We'll talk about those later in this presentation. We have atrioventricular valves, and this is the new school name, and I love this name. AB, or atrioventricular valves, are the valves in between atria and ventricles. So it's a very descriptive name that helps you to know where the valves of the heart are. Now, if you're interested in the old school names for the AB valves, they are the tricuspid and the bicuspid valve, and the old mnemonic was you got to try it before you buy it. The blood goes through the tricuspid valve before it goes through the bicuspid valve. But I'm a bigger fan of right AV and left AV because it's very, very, you don't have to think about it. The name tells you where it's located. So the right AV valve is in between the right atrium and the right ventricle. The left AV valve is between the left atria and the left ventricle. And I'd mentioned those fibrous muscles. Those fibrous muscles are called, well, if you're older like me, you call them chordae tendineae. And they are located right here. The new school name that's being phased in right now is tendinaceous cords or tendinous cords, but most people still say chordae tendineae. It really means the same thing. And these chordae tendineae connect to the papillary muscles, or those ridges of muscular tissue within the ventricles. And they also are going to connect to the flaps or the cusps of the AV valve. And the purpose of these chordae tendineae are to keep the AV valves from flipping out backwards. You can think of it kind of like a parachute. So we have the chordae tendineae, like the strings of the parachute. The cusps of the AV valve form the top of the parachute, and then the papillary muscle is the thing pulling down on the strings. In a parachute, it's a human being, and in our heart, we have the papillary muscles pulling on the chordae tendineae. And if you were to snap the strings of a parachute, the parachute would invert and no longer function. If we were to snap the chordae tendineae, the cusps of the AV valves would invert and we'd have blood flowing backwards. We also have semilunar valves. And most textbooks right now, I did a brief survey of textbooks over the summer, this is about a 50-50 split. Some textbooks will still use the term semilunar valve. Our textbook uses the term semilunar valve, so I'm going to keep using it and introducing it. Some textbooks, though, just drop semilunar entirely. The semilunar valves control the blood that is leaving the heart. They control blood flow out of the heart. And blood will leave the heart through two primary arteries. We have the aorta, or we have the pulmonary trunk. The pulmonary trunk has a valve known as the pulmonary semilunar valve. Some textbooks, though, will just drop semilunar and just call it the pulmonary valve because it's still very descriptive. Semilunar isn't necessarily necessary to know what you're talking about. And the same is true of the valve for the aorta. Our textbook says the aortic semilunar valve, but some other text will say aortic valve and just drop the term semilunar entirely. So if you're talking to another healthcare professional in a couple of years when you're living the dream and working at your job, and they say just aortic valve, it's okay. They mean aortic semilunar valve, even though in class we're calling it aortic semilunar valve. They're the same things. As we look at these semilunar valves, the cusps of these valves are very pronounced. A common congenital defect, which is a defect that somebody is born with, is that one of the cusps of the aortic valve is going to be defective and allow blood to flow backwards back into the left ventricle. The best man in my wedding had one of those defects, for instance, and he now has a mechanical valve installed in his heart. So, I'm just going to throw this out there. You must memorize the pathway of blood flow through the heart. You need to know every chamber, every major blood vessel going in and out, and you need to know every valve. And there's no silly rhyme associated with this. You just got to memorize it. When I was a high school teacher, I would take the back of my classroom and I would use masking tape, and make a big heart in the back of my room and make my students walk through it and say the name of the structures as they were walking through it. This is a good idea for study groups. We have way too many people in this room to do this in the front of the room. You can even buy shower curtains if you want that have this on it. And you can walk on the shower curtain. As we're looking at, I'm thinking for a more college-appropriate way to do it, maybe a better way as a college student to do this would be to just write the name of a structure on a note card and kind of play 52-card pickup, throw them on the ground, and then reorder them in pathway of blood flow through the heart. Now, since this is a circuit, a circular flow of blood throughout our body, we need an arbitrary starting point. My arbitrary starting point is always going to be the right atrium. And I just picked that arbitrarily. Now, as we're looking at blood moving through the heart and moving through the chambers of the heart, guys, gals, when a chamber contracts, blood is going to flow out of that chamber. When a chamber of a heart relaxes, blood will flow into that chamber. And this has to do with fluid flow rates. All fluids, whether it's a liquid fluid or a gaseous fluid, will always flow from an area of high pressure to an area of low pressure. I'll say that again because this is a fundamental idea behind the movement of fluids in our bodies. Fluids will flow from areas of high pressure to areas of low pressure. So when we contract a chamber of our heart, we increase the pressure on the blood in that chamber and make the blood flow out of that chamber of the heart. And when we relax the chamber, we lower the pressure and blood will flow in. So as a ventricle is relaxing, the pressure drops in the ventricle, and then the atrioventricular valves pop open, and blood will flow into the ventricles from the atria. When we contract a ventricle, the pressure increases, the AV valves snap shut, and then the pulmonary and aortic valves pop open, and blood will flow out of the heart. So if we look at our generic heart, you could just memorize these in order. I'm circling this, I'm putting a star next to this, I'm drawing an arrow next to this. I can't make it any more obvious. You need to memorize the steps of blood flow through the heart. I had to do it when I was in 6th grade. I know you as college students can do it. So as we're looking at the paths of blood flow through the heart, we're going to start with our right atrium. From the right atrium, blood will flow past the right AV valve into the right ventricle. From the right ventricle, blood will flow past the pulmonary valve into the pulmonary trunk, and then go through the pulmonary arteries to the lungs. While the blood is at the lungs, it's going to be dropping off carbon dioxide, picking up oxygen, and then going back to the heart through the pulmonary veins. Notice, guys, gals, and this is a common exam question, the pulmonary veins, even though they're a vein, they have oxygenated blood in them. And then the pulmonary arteries, even though they're an artery, they have deoxygenated blood. Next chapter we talk about blood vessels. I'll steal a little thunder right now. The word artery and vein refers to the direction of blood flow, not the oxygenation state. Arteries are going to always flow away from the heart. Veins always flow towards the heart. So when we look at the pulmonary circuit, the oxygenation state of the pulmonary circuit is going to be reversed compared to the oxygenation state of the systemic circuit. And since the systemic circuit is the biggest one, that's the one most of us think of. Back on track here, though. So we have the pulmonary veins. They carry oxygenated blood back to the left atrium. From the left atrium, we go past the left AV valve to the left ventricle. From the left ventricle, we're going to go past the aortic valve to the ascending aorta, then to the aortic arch, out to the systemic circuit. We'll talk about those blood vessels later. We deliver oxygen to the tissues of our body, pick up carbon dioxide, and then that deoxygenated blood will enter the heart through the inferior vena cava and the superior vena cava to go back to the right atrium. Let's talk coronary circulation. When we have coronary circulation, this is a subsection of systemic circulation. It's included within the systemic route because the blood supply comes off of the ascending aorta. Now, it's very important to oxygenate our heart. If we don't take care of the heart won't take care of us. So we need to supply blood to the heart to keep it constantly going. And what we find is that the heart is one of its own largest customers. 5% of the blood in our body goes to oxygenate our heart, even though our heart weighs less than 1 pound. So like for me in particular, I'm for easy math, we'll say 250 pounds. My heart is about 1 pound. My heart is less than 0.5% of my body mass, but my heart takes up about 5% of my total blood flow. So our heart is disproportionately greedy when it comes to blood consumption because it's going to be constantly burning through so much ATP because it's constantly working to pump blood. Our heart is going to use approximately 1/4 liter, or we'll say 250 mils of blood per minute. We need lots and lots of O2 in our heart and lots of sugars being delivered primarily so we can make ATP through aerobic respiration. We need to pump out lots of ATP. Now as we look at the two primary blood vessels that supply the heart, we're going to have the left coronary artery that comes off of the ascending aorta and extends anteriorly to the anterior interventricular sulcus. And we also have the right coronary artery. That right coronary artery is also going to come off of the ascending aorta, extend anteriorly, and move towards the right side of the heart. When somebody is having a heart attack, a lot of times there's some pain in their chest. Remember last semester when we talked about muscles, we have the pectoralis major, the pectoralis minor. Pain in the chest is referred to as angina pectoris, or pain localized near the pectoral muscles. And we find that this is primarily going to be associated with ischemia of the heart muscle or cardiac muscle. When we think of ischemia, I want you to think of reduced blood flow. So if we reduce the blood flow to the cardiac muscle tissue, that's going to induce pain in our chest. A common symptom of pronounced pain in the chest, or if somebody's really suffering from a heart attack, a lot of times that pain will be mirrored and manifested down the left arm of their body as well. So if you're working with a patient and they have tightness in their chest and their left arm hurts and they're breathing hard and they look like, they don't feel very well, those are giant red flags that they're in the middle of having a heart attack. As the myocardium is losing blood supply, that heart muscle tissue, the myocardium, is going to shift from aerobic respiration to anaerobic respiration. Anaerobic respiration means respiration without oxygen. So we're going to skip the citric acid cycle. We're going to skip the electron transport chain. We're only going to use glycolysis to generate a measly 2 ATP per glucose. And then we have lactic acid as a waste product. That acid builds up and helps to overemphasize the pain of the angina pectoralis. If this ischemia continues unabated, our heart cannot live very long on anaerobic respiration, and eventually the cardiomyocytes start to kick the bucket and die. When that happens, we have an infarction. Infarction referring to tissue death. What kind of tissue is dying in this situation? The heart muscle tissue, the myocardia, is dying. So a myocardial infarction is the death of heart muscle cells due to a lack of blood supply, aka it's a heart attack. Now, when we look at cardiomyocytes compared to traditional skeletal muscle tissue that we spent tons of time talking about last semester, in regular skeletal muscle tissue, we have one kind of tropomyosin. In cardiomyocytes, there's a different kind of tropomyosin. And as cardiomyocytes start to die, they'll lyse and release cardiac tropomyosin into the bloodstream and cardiac troponin into the bloodstream. And this is the result, and this is the basis of a standard blood test. If you are having a heart attack, or you think you may be having a heart attack, healthcare professionals will sample your blood and try to look for cardiac troponin, or the troponin specific to cardiomyocytes in your blood. And if it's found, that means that your heart muscle cells are in the process of actively dying when they took that sample. When we look at cardiomyocytes, it kills a lot of people in the United States. We've reduced deaths from most of the communicable diseases in the United States, but heart disease or heart-related diseases still are one of the biggest killers in the United States. As we look at the cardiomyocytes, or the heart muscle cells in our bodies, these heart muscle cells are going to be the in-betweeny weenies that we talked about when we talked about muscle tissues. So we had three kinds of muscle tissue we talked about last semester. We had smooth muscle tissue, we have skeletal muscle tissue, and in between we have the cardiomyocytes. Smooth muscle tissue is weak and can contract forever. Skeletal muscle tissue is super strong, but fatigues very quickly. And then in between, the cardiomyocytes, the heart muscle tissue, has reasonable endurance and reasonable contraction strength. It has a blending of the characteristics. When we look at the histology of a cardiomyocyte, we can see a little bit of striations, but not a lot of striations. Skeletal muscle is known for big, bold, and beautiful striations. Cardiomyocytes have mild striations. And if we look at this slide, you totally can't see that on the screen. I can see it on my computer screens because they're higher resolution, but we have very mild striations shown in those cardiomyocytes. Up on the screens, you just might be able to see the intercalated discs. I'm going to make them blue for us here. And those intercalated discs are only present in cardiomyocytes. And as we look at those cardiomyocytes, the intercalated disc is a key structure that allows for us to electrically coordinate the contraction of our heart muscle tissue, the cardiomyocytes. These intercalated discs have lots of folds in them that increase the surface area and maximize the interaction of one cardiomyocyte to the next cardiomyocyte. Those folds are referred to as interdigitating folds, or another way of thinking of them is the finger-like projections in between, digit referring to finger. So when we look at a cardiomyocyte, if we were to zoom in really close, we could see one cardiomyocyte on this slide on top. It has mild branching, and at the end of each branch is an intercalated discs. If we zoom in on an intercalated disc, we can see little bumps. Those little bumps are the interdigitating folds. And then there's some special structures also associated with those intercalated discs. We're going to have some mechanical junctions. When we think of mechanical junctions, I want you to think of that cell-to-cell connection that's very tight. It anchors those cardiomyocytes together. If the cardiomyocytes were not anchored together, while our heart was contracting, our heart would fragment itself. And since our heart is constantly contracting, we need very strong connections from one cell to the next cell to give us structural integrity to our heart. We're also going to have a layer of connective fibers. In this slide, the layer of connective fibers is the yellow layer, and it's referred to as the fascia adherens. And as the name implies, adherens tells us what it does. It adheres one cardiomyocyte to the next cardiomyocyte. Our fascia adherens is where we have those mechanical junctions localized. We're also going to have desmosomes. Those desmosomes are a slightly different kind of junction. And when we look at the desmosomes, they are going to have extensions into the cytoskeleton of one cell and then into the cytoskeleton of the adjacent cell. In this slide, you can see the desmosome. If you were to look at on your computer screen, probably not up on the monitor, the desmosome is right here, and then it has a bunch of fibers branching into one cell, and then it would also have a bunch of fibers branching into the other cell. And then personally, what I find to be the most interesting about the intercalated discs, and this is what most A&P teachers really emphasize, is the gap junctions, or the electrical junctions. I taught you gap junctions last semester when we covered this. Our gap junctions are perforations. They are pores or holes that allow the ions, the electrolytes from one cardiomyocyte to flow directly into the adjacent cardiomyocyte. In other words, these heart muscle cells are sharing cytoplasm with each other. And because they share cytoplasm, when one cardiomyocyte has an action potential traveling down it, that action potential will continue directly from one cell to the next cell to the next cell without having to use any neurotransmitters to go across the synapse. We refer to these as electrical synapse, as opposed to chemical synapses that we talked about last semester with all of the neurotransmitters. Now, when we look at our cardiomyocytes, I said this before, and I'm emphasizing on this slide, we need aerobic respiration. We need lots of oxygen, and we need lots of nutrients, ideally sugars supplied to the heart muscle tissue to make tons of ATP. Because our cardiomyocytes are really working so hard and they need lots of oxygen, we're going to have a lot of myoglobin present in the heart as well. Myoglobin is a special molecule that is 1/4 of a hemoglobin. So if you were to take just a generic hemoglobin molecule with its 4 subunits and chop those 4 subunits up so that we had 4 individual parts, each individual part would be a myoglobin. So a myoglobin will have one globular protein, it has one heme, and can store one oxygen gas molecule. Now, as we can imagine, myoglobin has iron and a dark coloration. And this is why, for those of you who have ever had eaten beef heart, you can buy that at the grocery store still. Beef heart, if you have like beef heart burritos, it has a very dark coloration to the meat. It's very dark meat and it's very iron rich. Now, our hearts also have lots of mitochondria within their cardiomyocytes because they need so many ATP. Now, because it's so important that our heart keeps pumping, our heart is flexible. It has the ability to use glucose, it has the ability to use beta oxidation of fatty acids, and it's also capable of going through gluconeogenesis and using amino acids to generate ATP. However, our heart can't come up with an alternative oxygen. All of these pathways of making ATP require lots of oxygen, so we need well-oxygenated heart muscle tissue. Our heart is also very resistant to fatigue because it makes very little use of anaerobic respiration, aka fermentation. It's predisposed to pumping out lots of ATP because it's so flexible in its fuel source for making the ATP. So let's have a review question. And I'm going to give you an extra 60 seconds on this question. Please arrange the steps in order of blood flow through the heart, from the superior vena cava to the aorta. And I did not list all of the steps. So you have to mix and match. You should talk to your partners. Probably one of you should flip back to the slide that shows the steps of blood flow through the heart. We have to mix and match these steps in order. Not all steps are shown. We're down to 60 seconds. Thirty seconds. Make sure you hit submit. You don't have to be right when you hit submit. Just hit submit so your answers get recorded. Ten seconds. So, guys, gals, when we look at the steps of blood flow through the heart, if we start at the superior vena cava, that superior vena cava drains directly into the right atrium. Let's show the answers here. From the right atrium, the blood goes past the right AV valve into the right ventricle. From the right ventricle, it goes past the pulmonary valve to the pulmonary artery. The pulmonary artery carries blood to the lungs. From the lungs, it goes through the pulmonary veins to the left atrium. From the left atrium, it goes past the left ventricle, past the left AV valve to the left ventricle. And from the left ventricle, it goes past the aortic valve to the aorta. So responses, apparently I can't see responses right now. All right. I'll figure it out later. Moving on, let's talk about electrical activity within our heart. The conduction of action potentials in our heart is a big deal. If we have inappropriate action potentials in our heart, we can have fibrillations, where our heart is just quivering. When our heart is quivering and we're having fibrillations, we need to use an anti-defibrillation or a defibrillation device to electrically shock the heart and reset its conduction system. That's the basis of those electrical pads you zap people with. Sometimes we can have tachycardia, sometimes we can have bradycardia. There's all different kinds of disease states associated with improper electrical conduction of the heart. Since we have a lot of nursing students in here, I'll just give you a heads up. It's common in nursing classes to have to spend a lot of time learning about the different kinds of electrical malfunctions in the heart. And there is a foolproof method for learning these malfunctions. I'm not going to spend a lot of time on them, but if you were to just stand in one spot and hold out your arms can represent the atria, and your legs can represent the ventricles. And while our heart is pumping, and this is a key concept, guys, both atria contract at the same time, then they relax, and then both ventricles contract at the same time, and then they relax. So a good heartbeat would be atria contracting, ventricles contracting. Atria, ventricles. That's a healthy heartbeat. Occasionally, you could have atria contract, ventricles contract. Wait a second. Atria contract, ventricles contract, and then ventricles contract. And you can skip the atria. That's a common disease. Or you can have a tachycardia, which is an elevated heart rate. You can have atria, ventricles, atrias, ventricles. Or that induces tachycardia when you do that. Or you could have a fibrillation. A fibrillation is an inappropriate contraction. Instead of a coordinated contraction, it's a quivering of the chamber of the heart. So to show it in the dance, you kind of go like this, and you just wiggle around. So we need to coordinate the heartbeat. And as we're looking at our heartbeat, it's all going to be based off of the pacemaker, or the sinoatrial node in our heart. That's going to coordinate our heartbeat. And just as a heads up, these next couple slides, this next section, it's pretty beefy. There's a lot of physiology in these next couple slides, because we're going to spend a lot of time about electrical activity in the heart. So when we look at our heart, we need to coordinate the heartbeat. As we're coordinating our heartbeat, our sinoatrial node, or pacemaker, is going to be the starting point. Our SA node is typically going to be located on the superior medial margin of the right atrium. And that's going to start the process. Let's switch colors here so green can stick out. Once the SA node initiates contraction, we're going to have an action potential travel to the atria, and both of the atria contract. That action potential will then also travel to another node. This second node is known as the AV node, or atrioventricular node. I'll spend some time on this photo for a while. From the AV node, the signal travels down the bundle branches, and then to the apex of the heart, and unfortunately my screen is cut off. And from the apex of the heart, with the left and right bundle branches, it heads upwards through the myocardium of the ventricles, and then will cause the ventricles to contract. Now, if everything in the heart was contracting at the exact same time, we would pressurize all of the chambers of the heart at the exact same time, and blood flow would be impeded. Blood will flow from areas of high pressure to areas of low pressure. And since the ventricles are bigger and beefier compared to the atria, the ventricles would overpower the atria, and blood would always flow out of the ventricles. It would never flow into the ventricles, and our conduction system, our blood circulation would shut down. So we need to time the contraction and relaxation of our heart. And the timing happens with the AV node. This is the pause button, so to speak. The AV node pauses the action potential. So the pacemaker node, the SA node, initiates an action potential. The atria contract. That action potential will then travel to the AV node, and at the AV node, it pauses for a fraction of a second. And while that action potential is pausing for a fraction of a second at the AV node, that allows the atria to contract, and the ventricles are still relaxed and will fill up with blood. If our AV node did not pause the action potential, atria and ventricles will contract at the same time. But thankfully, it pauses it. And as the AV node pauses that action potential, we'll wait a fraction of a second, and then the action potential continues on. It will then travel down the bundle of His, which is probably what Ms. Longdrich and I were taught, but the new school name is the AV bundle. And then we'll go to the left-right bundle branches. The term, and I've never seen this term until I looked at your edition of this textbook, is the subendothelial conducting network. But that's not fun to say. It's more fun to say Purkinje fibers. Purkinje fibers are those little fibers that send the electrical signal through the myocardium of the ventricles. The new school name is subendothelial conducting network. Blech. So, think back to our last unit of our last semester, the nervous system. Within the nervous system, we have the parasympathetic, we have the central and then the peripheral nervous system. Within the peripheral nervous system, we have the sensory and motor subdivisions. And then within the motor subdivisions, we have autonomic and voluntary. And then within the autonomic division, we have sympathetic and parasympathetic. When we think sympathetic, you think fight or flight, something that gets you ready for a lot of physical activity. When you think parasympathetic, you think rest and digest, something that helps you relax, recover, and recuperate. So when we look at our heart, the sympathetic branch of the autonomic nervous system is going to upregulate our heart rate. It's going to make our heart pump faster, pump harder. It's going to make us ready for a fight. And then the parasympathetic branch is the one that slows our heart rate and helps us to rest and relax. And during these steps, we'll have contraction and relaxation of the chambers. The technical term for the contraction of a chamber of the heart is systole. And then the relaxation of a chamber of a heart is dystole. And even though this is a generic term that refers to a chamber of the heart, in practicality, we really care about the left ventricle. Because when the left ventricle is going through systole, when the left ventricle is contracting, it's pushing blood through the aorta to the systemic circuit. And when the left ventricle is going through dystole, when it's relaxing, it's filling and blood is not being pushed through the aorta anymore. The systole and dystole of the left ventricle corresponds to the systolic and diastolic blood pressures, that 120/80 millimeters of mercury that are measured when you have somebody's blood pressure measured. For a typical resting adult, you will say average adult, there's a regular depolarization of the SA node or pacemaker. That gives us the sinus rhythm of the sinoatrial node, which for most adults is between 70 to 80 beats per minute. BPM is beats per minute in this context. This is also referred to as the vagal tone because this is the default rate of heart rate that's determined by the vagus nerve of the parasympathetic nervous system. So vagal tone gets its name from the vagus nerve. Now when we look at the pacemaker of the heart needs to depolarize on a regular, repeated rate. And it depolarizes at a regular, predictable rate because it has a lot of leak channels. And when we look at the electrical activity of pacemaker cells in the heart, they are going to be electrically different from the other cells of the heart, which allows them to coordinate the overall action potentials. So when we look at these pacemaker cells, they're going to have a constant influx of sodium because they have lots of sodium leak channels. And then once they hit negative 40 millivolts, we can think of that as threshold for the pacemaker cells. Calcium-gated ion channels are going to open up, and calcium and sodium will continue to rush in. Particularly calcium rushes in very quickly. As we add more 2 positive calcium ions to the inside of the cell, the voltage skyrockets all the way up to about +5. And at about +5 millivolts, or 0 millivolts, depending on how you draw a dotted line over, we are then going to close those potassium-gated channels, and we have sodium flowing out. So thankfully, this is the same as those motor neurons we covered last semester. Sodium rushes out, just like sodium did last semester. Voltage goes back down again. We reset everything at the hyperpolarized state, and then we are going to have our sodium channels slowly leaking sodium back in to get us back to threshold. And the amount of time from right here to right here is going to be dictated by that pacemaker potential. So that time lag dictates our cardiac rhythm. Question, comment? Potassium, did I say potassium flows out down here? Yeah, potassium flows out. That's what I meant to say. And if I said sodium flows out, I misspoke. Potassium, with a K positive, flows out. So as this SA node is depolarizing and reaching its threshold and initiating an action potential, it determines our heart rate. And when we think of a generic pacemaker or sinoatrial node, it depolarizes about every 0.8 of a second. And that corresponds to 75 beats per minute. You take 60 seconds divided by 0.8, and you get about 75. Now, the amount of time it takes the action potential to go from the SA node to the AV node is about 50 milliseconds, or 1/20th of a second. And then once that action potential gets to the AV node, it's going to be slowed down and paused for 100 milliseconds, or 1/10 of 1 second. So for those of you who don't like milliseconds, you could say 1/10 of a second. And that 1/10 of a second delay at the AV node allows the atria to contract and fill the ventricles with blood. After the AV node, the signal travels down the AV bundle through the interventricular septum to the apex of the heart. From the apex of the heart, that signal is then going to travel through the subendothelial conducting network, the Purkinje fibers, until the ventricular myocardium, the heart muscle cells of the ventricles, are going to be depolarized and all contract in a coordinated rhythm, or in unison. And as they are contracting, the contraction of our cardiomyocytes originates at the apex of the heart and then will travel superiorly through the ventricles. And this is going, if we look at the spiral arrangement or the cardiac vortex, this is going to cause the heart to twist and wring out the blood from the heart. This is a good graph to spend a lot of time on, both for lab and lecture. I'm going to put a star on this slide. So as we look at electrical behavior, and how are we doing on time here? We have 9 minutes left in class. So let's start at the top here. We're going to have some cardiomyocytes. And those cardiomyocytes are going to have sodium influx, sodium flowing in until they reach threshold. At threshold, our myocardium, or heart muscle cells, will depolarize and will have sodium inflow that will open sodium voltage-gated channels and cause the voltage on those cardiomyocytes to skyrocket. And it'll peak here at about, it looks like 29, 30-ish millivolts. And then our sodium voltage-gated channels close. And at this point in time, if we look at a heart muscle cell, we're going to have the plateau stage. So when we thought of a motor neuron and when we thought of a skeletal muscle cell, there was a big up peak and a big down peak. Cardiomyocytes have a sharp up and then a gradual plateau at an elevated voltage. This plateau phase, as we look at the voltage of our cardiomyocyte, is going to have calcium ions slowly moving across the membrane. And this slow flux of calcium ions is going to prolong that depolarization phase. But eventually, that voltage keeps ticking down, down, down slowly, and eventually, as more and more potassium goes out, our potassium channels are going to continue to remain closed until we reach this approximate voltage. And it's not an on-off, it's a gradual transition. At this stage, step number 5, we are going to have the calcium channels close. Calcium is going to be pumped out of the cell, and then but the potassium channels open out, and we have the giant outflux of potassium. So potassium ions rushing out. So this graph is kind of complicated. The green line on the graph refers to the voltages of a typical heart muscle cell, a cardiomyocyte. So when we look at an action potential of a cardiomyocyte, there's a sharp uptick, a plateau, and then an exponential decline to a logarithmic bottoming out. And then in red, we have the contraction state. And I really wish that there was 2 y-axes. One y-axis over here in green for the voltage, and another y-axis over here in red that had something like kilopascals, or actually pascals, or some kind of a unit of force, maybe millinewtons, to show the contractile force of the cardiomyocyte. So during this action potential of the cardiomyocyte slowly contracts, peaks, and then starts to relax again. So the cardiomyocyte begins to relax about the same time that the potassium voltage-gated channels pop open. And we have that rapid potassium outflux from the cardiomyocyte. There's something else that's pretty notable on this slide, and that is the absolute refractory period of a cardiomyocyte. During that absolute refractory period, think back to Unit 4 of last semester, the nervous system. During the absolute refractory period, you cannot initiate another action potential regardless of how strong your stimulus is. This absolute refractory period dictates a theoretical maximum for your heart rate. And when you look at the theoretical maximum heart rate, as dictated by the absolute refractory period, it's approximately 25-ish 250 milliseconds, or about 2.5 tenths of a second. That translates to about 230 beats per minute as a physiological max heart rate. I'm in my 30s, so my max heart rate right now, when I go in the Stairmaster at the YMCA and put my hands on those sensors, I max out at about 190 beats per minute now. I can't pass 200 anymore. If I pass 200, I'm toast. But teenagers, 15, 16-year-olds that are huffing and puffing, they can get above 200 beats per minute, no problem. As we age, this absolute refractory period is extended, and our max heart rate goes down. When we're looking at the max heart rate of our heart, and just the electrical activity of our heart, we can look at an ECG or an EKG. The K right here, lots of times students will ask me, why is it an EKG as well? That refers to the German word for heart. So in English, it's ECG, but if you were to use German words, it would be EKG. And when we look at an electrocardiogram, our electrocardiograms are going to take all of the action potentials of all of those cardiomyocytes, of all of those neurons, all of those axons of the neurons, and combine them all together. The first EKG was recorded in the late 1800s. Let that sink in for a moment. Electrocardiograms were first recorded before telephones, before automobiles, before airplanes. Most people were still using sticks and rocks in their house as common tools. And then some guy in the city was able to record an EKG. That's very impressive to me. Now, granted, the EKG was super primitive, and it didn't have all these nice bumps, peaks, and curves. It was just a blip, blip, up and down, and then an up and down. The instrumentation was not very sensitive. Another common question I get from students is, why are the peaks and valleys and all those divots in the EKG labeled P wave, QRS, and S and T, and all? Like, why those letters of the alphabet, as opposed to like step 1, step 2, step 3, or maybe A, B, C, D? And the reason for this is the individual who performed the first EKG had the foresight to recognize that his or her instrumentation wasn't that good. And this individual believed that as the instrumentation improved, they may find more bumps or divots at the beginning or more bumps or divots at the end afterwards. So they chose a letter of the alphabet that was kind of in the middle. And to really emphasize this, they called their initial bump the QRS complex. As we look at that QRS complex, that in particular, the R peak, is the big upturn in the EKG. And we only have time for this slide, then we'll be done. So let's talk P wave. During the P wave, this P wave corresponds to the SA node firing and the depolarization and contraction of the atria. And then the signal gets to the AV node. From the AV node, goes down the bundle branches to the Purkinje fibers. And while our atria are repolarizing and the ventricles are depolarizing, lots of crazy things are happening. Because the ventricles are so ginormous, when they depolarize, that generates a lot of electrical activity and gives us the big spike in the QRS complex. The ST segment is going to correspond to a slight delay. And the timing of that ST segment can be used to predict cardiac disease. And then after the ST segment, after that slight delay, after we contract the ventricles, we wait a fraction of a second, the blood flows out of the ventricles, and then we relax the ventricles. And that relaxation of the ventricles corresponds to the T wave. Make sure for lecture exam purposes, you know contraction, relaxation, depolarization, and repolarization for each part of an electrocardiogram. And that's all we have for today.